We are organisms. We are conceived, we are born, we live, we die, and we decay. But as we decay, we feed the world of living plants and bugs and bacteria. Welcome to Econdon Development Center, Majalis Arts and Science College, Pramanur. I am Naren Ayub, Department of Microbiology. Today I would like to present the ultrastructure of bacteria. At the end of the video, you are able to identify the structures and functions of bacterial cell, and you are able to understand that the prokaryotic cells have some features in common with the eukaryotic cells and their differences. So let's begin. Bacteria are unicellular organisms which have a variety of sizes, shapes, and structures. Ultrastructure of bacteria includes both external structures as well as internal structures. So let's begin with the external structures. Some prokaryotes have a layer of material outside the cell wall called Glycogalyx. This layer has different names depending on its characteristics. Capsule is a Glycogalyx layer composed of firmly associated polysaccharide molecules within the cell wall, which is difficult to be washed off. Whereas slime layer is also a Glycogalyx layer composed of loosely associated glycoprotein molecules, which can be easily washed off. Now let's move on with the functions of capsule. As we all know, capsule contains a lot of water. It prevents the bacteria from desiccation. It prevents the bacteria from phagocytosis. It helps for the attachment of bacteria to the host cell. S layer. Some prokaryotes possess S layer, which is present outside the cell wall. In gram-negative bacteria, S layer occupies directly on the outer membrane. In gram-positive bacteria, it is associated with the peptidoglycan. It helps to maintain the shape and rigidity of the cell. It helps to protect the cell from pH fluctuations, osmotic stress, enzyme, and phagocytosis. Now let's discuss about flagella. Flagella is the thread-like appendages extending outward from the plasma membrane and cell wall. Flagella is a rigid structure about 20 nanometers across and 15 micrometers long. It consists of three parts: the filament, hook, and the basal body. The filament is the hollow, rigid cylinder, which is composed of protein subunits called flagellin. Its molecular weight ranges from 30,000 to 60,000 daltons. Flagellar hook is the broader region, which links the filament to its basal body and is composed of different protein subunits. Basal body is the complex part of flagella. In gram-negative bacteria, basal body consists of four rings. The outer L and P rings are associated with the lipopolysaccharide and peptidoglycan layer. The inner M ring contacts the plasma membrane. The innermost C ring is associated with the cytoplasm. Gram-positive bacteria possess two basal body rings. The inner ring connects the plasma membrane and the outer ring connects the peptidoglycan. These two rings are composed of proteins like mode A, mode B, and Flag. Here you can see the structure of flagella. Now we have Fimbri and pili. Fimbri are hair-like appendages that are thinner and shorter than flagella. A cell may be covered with up to 1,000 Fimbri. It helps for the attachment of bacteria to the solid surface. Some bacteria possess 1 to 10 sexpili, which differ from Fimbri as they are larger than them. Sexpili functions for conjugation. Conjugation is the process of transformation of genes from one organism to the other. Now we have cell wall. Cell wall is the layer which is present outside the plasma membrane. It helps to maintain the shape of the cell. It protects the cell from osmotic lysis. It can contribute pathogenicity to the cell. Cell wall is composed of two subunits like glycan portion and peptide portion. Glycan portion is composed of two alternative derivatives like N-acetylmuramic acid and N-acetylglucosamine. Peptide portion is composed of four amino acids like D-alanine, D-glutamine, L-alanine, and L-lysine. Peptide portion of four amino acids are linked to the carboxyl group of N-acetylmuramic acid to form peptidoglycan subunits. Each peptidoglycan subunits are interlinked by the peptide interbridge between two peptides to form cell wall. Based on the difference between cell wall, bacteria are classified into gram-positive and gram-negative cell wall. So let's dive into the difference between gram-positive and gram-negative bacterial cell wall. Gram-positive bacterial cell wall is composed of thick, homogeneous peptidoglycan layer, whereas gram-negative bacterial cell wall is composed of thin heterogeneous peptidoglycan layer. Here the cell wall contains two layers like outer membrane and peptidoglycan. Gram-positive bacterial cell wall is more resistant to the osmotic pressure than gram-negative bacterial cell wall. Gram-positive cell wall contains teichoic acid, which is covalently bonded with the peptidoglycan, whereas gram-negative bacterial cell wall lacks teichoic acid. Here you can see the structure of gram-positive and gram-negative bacterial cell wall. Now, gram-negative bacterial cell wall is more complex one because it contains outer membrane, which is composed of lipopolysaccharide and consists of three parts, like lipid A, core polysaccharide, and O antigen. Lipid A functions as hydrophobic domain, which is the endotoxin and the main virulence factor. Core polysaccharide contribute negative charge to the cell surface, and O antigen shows versatile nature in order to protect the cell from host defense. Cell membrane serves as selectively permeable barrier. It acts as the location for crucial metabolic processes like respiration, photosynthesis, and so on. The fluid mosaic model of the membrane is discovered by Singer and Nicholson. They propose that the membrane is the lipid bilayers within which the protein floats. Cell membrane is the thin structure, about 5 to 10 nanometers thick. The membrane-associated lipids contains two ends like polar and nonpolar ends. The polar ends interact with water and are hydrophilic and are exposed, whereas nonpolar hydrophobic ends are insoluble in water and are buried interior. Two types of proteins are identified within the cell membrane. Peripheral proteins, which are connected loosely to the membrane and can be easily removed, and constitute about 20 to 30% of the membrane proteins, whereas integral proteins constitute about 70 to 80% of the membrane proteins. Hence, they are not extracted easily from the membrane. Bacterial membrane contains sterol-like molecules called hopanoids. Plasma membrane infoldings are common in many bacteria, and they may aggregate to form spherical and flattened vesicles called mesosomes. Their function is to provide larger membrane surface for greater metabolic activity. Now we have internal structures. Cytoplasm is a gel-like fluid inside the cell. It provides a platform upon which other organelles can operate within the cell. Ribosomes are complex structures made up of proteins and RNAs. They are the sites for protein synthesis. Cytoplasmic ribosomes synthesize proteins destined to remain within the cell, whereas the plasma membrane ribosomes make proteins for the transport to the outside. Now we have inclusion bodies. Inclusion bodies are used for storage. Some inclusion bodies live freely in the cytoplasm, while others are enclosed in a shell. Now let's dive into the organic inclusion bodies. So here we have glycogen. Glycogen is the polymer of glucose units formed by alpha-1,4 glycosidic bond, and their branching chains are connected to them by alpha-1,6 glycosidic bonds. Now we have poly-beta hydroxybutyrate. It contains beta hydroxybutyrate molecules which are linked by ester bonds between carboxyl and hydroxyl groups of adjacent molecules. Glycogen and PHB inclusion bodies are carbon storage reservoirs as they provide material for energy. Next we have carboxysomes. Carboxysomes are present in cyanobacteria and other carbon dioxide fixing bacteria. It contains enzyme ribulose 1,5-bisphosphate carboxylase, which is used for carbon dioxide fixation. This enzyme possesses a paracrystalline arrangement within the carboxysome. Gas vacuole. It provides buoyancy to the aquatic prokaryotes. It is present in the photosynthetic bacteria. They may aggregate to form small hollow cylindrical structures called gas vesicles. Gas vesicle walls are composed of proteins which forms an enclosed cylinder impermeable to water and are freely permeable to atmospheric gases. Now we have inorganic inclusion bodies. Some bacteria store phosphate in the form of polyphosphate granules or volutin granules. They function as the storage reservoir for phosphate. Polyphosphate granules are also known as metachromatic granules as they show metachromatic effect. Sulfur granules act as the storage reservoir for sulfur. Magnetosomes. It contains iron in the form of magnetite. The bacteria which possess magnetosome are called magnetotactic bacteria. The next internal structure is the nucleoid. Nucleoid is the irregularly shaped region where the genetic material is located. In prokaryotes, DNA is looped and coiled with RNA and a variety of nucleoid proteins. In addition to the genetic material present in nucleoid, prokaryotes contains extrachromosomal circular DNA molecules called plasmids. They are capable of self-replication. Now let's discuss about the types of plasmid. F factor. It contains genes that direct to the formation of sex pili. R plasmid. It contains genes that encode for the enzymes which are capable of destroying antibiotics. Called plasmid contains genes for the synthesis of bacteriocins. Bacteriocins are the bacterial proteins that destroy bacteria. Virulence plasmid. It encodes for the factors which makes their host more pathogenic. Metabolic plasmid. It carries genes for the enzymes to degrade the substances such as pesticides. So that's the end of the session. Thank you. The PDF of the presentation is given in the description box. Thank you.